Good morning, good afternoon, and good evening, everyone, and welcome to the first webinar in the Cardiovascular Connections 2022 series. Today's webinar is titled Cardiovascular Physiology in the Setting of Spinal Cord Injury, featuring Dr. Chris West, Associate Professor of Medicine at the University of British Columbia. Today, he'll be discussing his research that investigates the cardiovascular and autonomic changes that occur following spinal cord injury, as well as the efficacy of neurotherapeutic interventions. Before we get started, I just want to acknowledge our partners at the American Physiological Society and the European Council for Cardiovascular Research, and a special shout out to our session sponsor, Translational Systems, for helping to make this event possible. Translational is a company that is known for vascular blood flow, blood pressure, and ventricular pressure volume loop technologies in the life science field. Recently, they launched the world's first telemetric blood flow and blood pressure technology for rodents. Translational is proud to count today's speaker, Dr. Chris West, as one of their clients and key collaborators. And so, without any further delay, I'm very pleased to welcome Dr. Chris West to the floor. Chris, you can take it away whenever you're ready.
Thank you so much, Sarah, and thanks everybody for joining us. It's my distinct pleasure to be here today to launch this series and share some of the work that myself and the individuals in my lab have been working towards over the last 4 or 5 years, really around this setting of cardiovascular and autonomic function in the setting of spinal cord injury. So with that, what I want to do today is kind of take you on this bi-directional translational journey, sharing some of our findings from the bench all the way to the bedside and back again. I'm really trying to illustrate how we use the clinical setting of spinal cord injury to really probe some of our more preclinical or mechanistic studies. I have broken the talk up today into 6 or 7 distinct areas, so feel free to tune in and out to those, as some may be of more interest to you than others. We'll start just by giving a brief history of the field of spinal cord injury, because I appreciate this is a field that some may not be familiar with. I'll then briefly touch on my research program here at the University of British Columbia, and then following that move through really some of the key parts of this, which is looking at how the human heart responds to spinal cord injury, and then digging into some of the animal models we use, as well as some of the acute and chronic interventions. And then I'll just finish by summarizing really, what I see some of the future visions for the field are. So with that, I'll get started and really just giving a brief overview of the history of spinal cord injury. And, the field of spinal cord injury has come a long way. This is, one of the first real findings from the field and real notes about someone with spinal cord injury. And we've really progressed from this idea that spinal cord injury is an ailment not to be treated to really having now a multimodal healthcare delivery around optimizing quality and quantity of life. From here, really the first spinal unit was actually in the UK in Stoke Mandeville, and it was launched by Professor Ludwig Goodman, who's a real giant in the field of spinal cord injury. And what we see now is many institutions and specialized research centers around the world dedicated to this topic. But one of the interesting things is, and this is really seen in the names of these institutions and centers. So for example, Miami Project to Cure Paralysis, International Collaboration on Repair Discovery. And it's really this, pervasive imagery of how people see individuals with spinal cord injury and the names of these centers that has really driven the research area, which has largely been focused around, the paralysis that accompanies spinal cord injury. And so much of our preclinical or basic research has really focused on, a better understanding of the cellular response to injury in the spinal cord, and trying to understand how various types of cell therapies can be used to improve motor function or sensory function outcomes. We're now in a setting where we are increasingly using different types of technology to modulate circuitry below and above the injury with a view to regaining, again, motor and sensory function. But whilst motor and sensory function are some of the most, obvious to the eye for people with spinal cord injury, there's actually changes to a number of other body systems. And, we've known this for some time. So if we go back to some of the early research conducted in that Stoke Mandeville unit, what I'm showing here is a number of squiggly lines essentially, but this is how different physiological indices respond to bladder distension in an individual with cervical spinal cord injury. And this is, at least to my knowledge, really the first publication that looked at autonomic function after injury. And what you can see here is blood pressure tends to be quite low in this individual. And then with that first vertical line is the onset of bladder distension. And you can see, just if we take a look at peak blood pressure, there is this really high hypertensive response to this stimulus, something that we now term in the field autonomic dysreflexia, and this has been very well studied. But this certainly indicates that, there is something aberrant or different happening in these autonomic circuits after injury that can produce cardiovascular responses such as these. And I think if we take a look over the autonomic nervous system, this is just a classic depiction that you would see in a textbook. One of the important things to remember is autonomic nervous system obviously has two sides: parasympathetic, which is our rest and digest, and our sympathetic, which is kind of our activate nervous system. And the important thing to take away from this slide is where the innervation originates from these two systems. So the parasympathetic nervous system, these are largely cranial nerves. And so the parasympathetic innervation to our various organs is spared with most types of spinal cord injury. However, if you have a spinal cord injury in this cervical region, which is where around 50 to 60% of spinal cord injuries occur, what happens, depending on the severity of that injury, is that the brainstem control over the sympathetic nervous system becomes disconnected. So what that results in is these circuits below the injury essentially being unregulated or dysregulated. The important thing to note here is these circuits remain intact. They don't degenerate. They just change the way they function after injury. And it's really this disconnection from the brainstem that we're going to focus on a lot today. Of course, the sympathetic nervous system innervates pretty much every organ throughout the body. So you can imagine, in addition to sensory and motor dysfunction, there's really some quite profound autonomic changes that happen when the injury is in this cervical spinal cord region. So we're certainly not alone in this, and interest in the autonomic field is growing after spinal cord injury. I'm just going to highlight this with some simple, PubMed keyword searches, where we can see now we're up to about 100 papers a year, but still only 100 papers a year looking at spinal cord injury and autonomic function. If we compare that to the motor system, we're outweighed about 10 to 1 on the autonomic side. So there's really still a lot to learn. And, these studies date back to the early or the late 1800s. Here we're the mid 1900s before these studies get started. So there's a lot to learn in this area. And that's really kind of what stimulated, my interest and will to set up a research program in this area. So with that background, I thought I would just cover, and maybe this is more for the trainees out there, but how I went about developing this translational program of research to study the cardiovascular and autonomic responses. Many people say to me, "Oh, this is a very niche area. How did you get into it?" And this is one of the first, and really one of the ways I got into this. What you're seeing here is some simple heart rate recordings. These were collected about 15 years ago in the build up to the Beijing Paralympic Games. Each line represents a different individual, and these individuals were tasked with doing a 40-minute peak effort in their wheelchairs. And what you can see here, certainly in at least 2 of these individuals, is heart rate really does not get above 100 beats per minute, despite putting in a maximal exercise effort. And so, this was at least surprising to me in the first instance, and it got me thinking, wow, what would my exercise response be like if my heart rate didn't really increase as I went about exercising? Of course, as I went to the literature, this isn't the first time this has been shown. It's actually been shown many times. What I'm showing here is some findings from Maria Hopman's group. But what really this shows is if you take individuals with a cervical spinal cord injury, i.e. tetraplegics, if you look at either cardiac output or stroke volume with increasing exercise intensity, there's really no increase in these tetraplegic individuals, but there is in individuals that have a low thoracic injury. And so this really led me to the research question of, well, why is the stroke volume and heart rate response to exercise so constrained in this population? Fast forward a few years, and this really was some of the foundations for my research lab, which now looks at essentially the chronic and acute cardiorespiratory and autonomic consequences of spinal cord injury. We're fortunate to do this across 2 locations, one at that research center, the International Collaboration on Repair Discovery in Vancouver, and then also as the recently developed Center for Chronic Disease Prevention and Management, also at UBC in Kelowna. And the way we go about answering this, and some of the research I'll show to you today, is right across from using rat and pig models to try and drive some mechanistic and clinical testing through to the patient population. And then I've also done a reasonable amount of work with elite athletic population as well, who certainly represent probably the pinnacle of fitness that one can attain after spinal cord injury. I'm very fortunate along the way to have had really wonderful trainees and staff working in my lab, and I'll do my best as I go throughout this presentation to highlight some of their work as well. So with that background, I thought I would start by really trying to answer a straightforward question. This presentation will focus mostly on the cardiovascular and autonomic side of things, and we're going to leave the respiratory side for another day. So, first off, just how does the human heart respond to spinal cord injury? Now, spinal cord injury itself, there's about 2.5 million people living with spinal cord injury, about 500,000 new injuries a year. But despite that, compared to many other pathologies, the incidence and prevalence of spinal cord injury is quite low, which can make recruitment to study these individuals quite challenging. So to get around this, Alex Williams, one of the postdocs in my lab, recently conducted a meta-analysis that basically took every echocardiography study ever conducted in the field of spinal cord injury and looked to compare what happens in individuals with spinal cord injury versus age-matched uninjured controls. And this schematic kind of highlights the main findings. And what we see is that after spinal cord injury, there's really quite significant reductions of about 20% in left ventricular volume, left ventricular dimensions, and left ventricular mass. One of the interesting things for me, at least when I started in this area, is that ejection fraction, which is, our clinical measure of contractility, is actually unchanged after spinal cord injury. Why is this happening? Sometimes those mechanisms are hard to answer. And one of the other things with echo is, whilst it's great to give us measures of structure and function, it does have some underlying assumptions. And one of those is that all of the metrics that you can derive are dependent on loading conditions to the heart, which we now know change after spinal cord injury. So to try and understand some of these pathways better, the first step we took was to actually make sure that what we're finding in the echo meta-analysis was actually confirmed in an independent sample. So here I'm just highlighting a study which we recently completed that Shane Balthazar, a PhD student in Dr. Krasikov's lab. We did some imaging where we took cervical spinal cord injured individuals in the acute setting and subacute setting after injuries. That's within the first 5 months. And then we compared their structure and function of the heart against individuals that were chronically injured. What I'm showing in this first panel here is just example echo images from the left ventricle. And I think you can appreciate here already that the size of the left ventricle here in the chronic individuals is smaller. We made these measurements in around 50 individuals altogether. And that's indeed what we find, that not in the subacute, but in the chronic setting, there is this reduction in cardiac volumes. We took this a step further to look at the mechanical response of the left ventricle. Of course, the heart doesn't, just beat in space. It also twists and rotates as it is beating. And so using some speckle tracking echocardiography, we can indeed look at twist and mechanics. And what we find is these are ensemble averaged responses across the group. There is this reduction in twist, so the mechanical response is attenuated. And if we just quantify our peak systolic twist velocity, that is also reduced in these chronic individuals. So certainly some good evidence here from a prospective trial that the echo results do hold up. But again, many of these responses are dependent on loading conditions. And so one of the things that I was interested in was to try and develop some preclinical models. And this is really the work of two individuals in the lab, Erin, who's been my long-term lab manager, and Lisa, a PhD student with me. We've been interested to develop rodent models for spinal cord injury cardiovascular research. The rat model, what we now do is use a midline contusion injury. So this involves dissecting down to the T2, T3 spinal segment. And we do a weight drop contusion injury onto the spinal cord. We then recover these rodents for various amounts of time and study their cardiovascular and autonomic function. The important thing to note here is if we just look histologically above at the epicenter and below the level of the injury, is that these are not anatomically complete injuries. There are pathways that are spared through the injury, which replicates what happens clinically. It's just, how much function is spared in these pathways and what degree of cardiovascular function does one present with. The rodent models are great for testing chronic and behavioral interventions, but of course the size of the rat and from both a heart and spinal cord perspective is not the same as a human. And so in collaboration with Brian Kwan's group at UBC, we've also developed or extended the development of a pig model of spinal cord injury. This existed for the low thoracic region, but we've now extended this to also be a T2 level spinal cord injury. So we now have both a small and large incomplete high thoracic model of spinal cord injury. In using these models, we've also tried our best to develop clinically relevant outcome measures that enable us to take the findings from rodent and pig studies and move them towards the clinic. And so we use small and large animal echocardiography, where we can get the same views of the heart over time. The advantage, of course, with the rodent and the pig models is we can do some more invasive physiology measures as well. So a lot of our data that I'll share today is from left ventricular pressure volume loops using an LV pressure volume admittance catheter, which enables us to get volume and pressure measurements, but it also allows us to manipulate the circulation and calculate something called our end-systolic elastance, which is a load-independent measure of contractility and is kind of considered the field gold standard for assessing contractile function. More recently, we've also been interested in understanding how neural activity in these pathways relates to end organ function. And so we now also do direct sympathetic nerve activity recordings from both renal and splanchnic nerves, sometimes in isolation, sometimes together. And we can get these signals at the same time as our blood pressure and cardiac signals as well, to really understand how the sympathetic nervous system changes after injury and how these changes impact the heart and blood vessels. So, with the development of those injury models in mind, what have we learned from these injury models about the physiology of spinal cord injury that perhaps we didn't know from the clinical situation? And so I'm going to share some recent work here from looking at nerve activity and some blood pressure recordings in a cohort of rodents with spinal cord injury. And this has really been the work of one of my PhD students, Mehdi, in the lab. What I'm showing here is just some raw example recordings. In blue is the blood pressure, and in green is the sympathetic nerve activity in the splanchnic nerve. I'm going to keep the axis the same here and show now what happens after spinal cord injury. And so I think this immediately identifies two things. One, there is this clear reduction in arterial blood pressure with spinal cord injury. This is not an acute injury. This is a chronically injured situation. And that the amount of activity in the sympathetic nervous system is considerably reduced. It's still there. There is definitely activity in this circuitry, but it's just not to the same magnitude as it was before. We can do some characterizations of this and look at some of the bursts of sympathetic activity. And the one that really sticks out here is if you look at the area of these bursts, it is markedly reduced after spinal cord injured rats. And this is associated with quite profound reductions in systolic and diastolic blood pressure as well. What about in terms of the heart? So using echocardiography, we conducted a similar study in rats as to what we did in humans, except here in the rats, we were able to longitudinally track how cardiac volumes and structure changes with time in the same animal using echocardiography. Here's some example parasternal long axis. This is a sham injured rat on the left-hand side. And then this is with time post-spinal cord injury. And what you can see, again, similar to the human setting, is that the size of this left ventricle is changing with injury. And if we go on to quantify some of this work, which is the work of Mary Fossey for her master's in my lab, we can see spinal cord injury is associated with this gradual and sustained reduction in cardiac volumes. So again, this kind of mimics what we're able to see clinically. These volumes aren't changing immediately after injury, but with time, there is this reduction in heart size and function. Now, what about our invasive data? So in separate cohorts of rats in this study, we essentially exposed these rats to the T3 spinal cord injury. We then did invasive pressure volume catheterization to get left ventricular function, including IVC occlusions, which allows us to reduce blood flow back to the heart and make that measure of end systolic elastance, which is kind of the gold standard measure of cardiac contractility. Now, I'll recall that in the clinical data and indeed in our animal echocardiography data, we found no difference in ejection fraction. But if we look at the pressure volume data and quantify end-systolic elastance, and this has been done by many people in the lab, first really shown by Jordan Square, a PhD student, and I think this is some of the most clear data identifying cardiac function after injury. And what you can see is at 1 day, 7 days, 8 weeks after injury, just the size in the height of this pressure volume loop, it's reduced by about 30%. So there's about this 30% reduction in pressure generating capacity after injury. This is sustained into the chronic setting. And again, here you can see the gradual reduction in volumes. The width of this pressure volume loop is wider at 1 day and gets less with time post-injury. But what's really interesting is in response to these inferior vena cava occlusions, you get this left and downward shift of the pressure volume loop. If we plot this linear relationship on the left-hand side, this is our end systolic elastance. And there's a clear, almost 3 times reduction in cardiac contractility after injury. And this is sustained into the chronic setting. So again, unlike ejection fraction, end systolic elastance is not dependent on loading conditions. And so when you take out these changing loading conditions, we now have some clear evidence that spinal cord injury impairs cardiac contractile function. What about at the level of the cardiomyocytes? So we've also done histological examination of the cardiomyocytes. We've done this in 2 places. First, by taking some samples from the left ventricular myocardium and also the subepicardium, so we can get differentially orientated cardiomyocytes. And this allows us to make measures of both length, width, and cross-sectional area using quadruple immunofluorescence staining. Here's some examples from a sham injury, and here from an 8-week spinal cord injured rat. And the way we've decided to go about quantifying this is rather than just measuring 5 or 10 and taking the average value, we're now actually measuring 80 individual cardiomyocytes per animal and displaying this as a frequency histogram. And if we do this, I'm just showing data at 5 days, 7 days, and 8 weeks. I appreciate these are a little small, but what you can see is across all of our indices, whether it's length, width, or cross-sectional area, there is this gradual leftward shift in cardiomyocyte size, suggesting that there is actual evidence of cardiomyocyte atrophy occurring with time post-injury. And it's probably this reduction in cardiac atrophy that is driving some of these reductions in structure and function that we're seeing. What's the underlying cause of this? Well, I don't have time to share a lot of data with you on that today, but we recently published a paper basically implicating that it is actually the impaired descending sympathetic projections down the spinal cord that is responsible for this reduction in cardiac contractile function. I'll just share one piece of data from this paper, which is also included on the resources tab for anyone interested, that I think shows this the best. And so what we're doing here, this is some work originally of a postdoc in the lab, Mali. I'll try and orientate you to the figure. So we have our timeline here. This is going to be left ventricular catheterization before injury. We then do an acute T3 spinal cord injury. We continually measuring left ventricular pressure throughout this period. And then following about 2 hours of spinal cord injury, we also deliver intravenous hexamethomium bromide, which is a pan-ganglionic blockade. So what this is going to do is block any ganglionic transmission to the left ventricle. And if we just zoom in on some of these areas, we see here the left ventricular waveform of pre-injury. There's a clear reduction post-injury. And if we further deliver hexamethomium bromide, there's really no further reduction in cardiac pressure generating capacity. And this is really quantified in these next 2 figures here, looking at either left ventricular maximum pressure or DPDT max. So what this suggests to us is that at least some of the underlying cause of this left ventricular or reduction in left ventricular function is due to impaired sympathetic transmission. So now we've kind of highlighted the timeline and potentially one of the underlying causes. I'm going to switch now in these next 2 parts to talk about what we can do to treat cardiovascular dysfunction. Again, focusing mostly on the heart and blood pressure. I'll first highlight some of our studies in the acute and then move on to the chronic setting. So really in the acute setting, most of the research, as I said before, at least the basic science level is focused on cell therapies. despite many clinical trials, we're still really not there yet with delivering cell therapies clinically in the field of spinal cord injury. So that really leaves only two possible treatment options that can offer some form of neuroprotection. One is to decompress the injury. So here I'm showing some imaging on the right-hand side of a cervical injury. So we can decompress or remove the compression that the vertebra is placing on the spinal cord. And the second really is appropriate hemodynamic management. Now, why do we think hemodynamic management might be important in this acute setting? What I'm going to try and do is highlight this here. It's shown in blue now is what the initial trauma that was caused by the spinal cord injury is. But when this injury occurs, it sets off a whole secondary cascade of signaling mechanisms, including some profound hypoxia in the spinal cord. And this initial injury actually gets exacerbated with time. And so now the secondary injury really infiltrates, the spinal cord above and below the area, and you can get edema and bleeding in this cord, which sets off some cytotoxicity. And really, you then get this expansion of the injury site, which has a larger impact on impairing sensory and motor function sometimes than the initial trauma. And so really the idea in the field has, the thought in the field has been, well, if we can appropriately maintain blood pressure in the acute setting or increase blood pressure in the acute setting, maybe we can deliver more blood flow and oxygen to the spinal cord and prevent some of this secondary ischemic injury from occurring. This has been studied in a number of settings. I'm going to share probably, at least to my knowledge, one of the most comprehensive studies on this topic, which was done in individuals with spinal cord injury. In this study, as you've seen, blood pressure declines quite rapidly after injury. So this group here, which is some of the research in the States that's been performed, is to actually use vasopressors to increase mean arterial pressure and maintain it at a value of 85 to 90 for 7 days. What this group did, which is a bit different, is they actually had continuous recording, and they extracted these 1-minute recordings from 100 patients, resulting in over 1 million data points that they were able to actually analyze. What they essentially showed is if you then take these individuals and look at which individuals have the greatest recovery of function after injury, so that's shown here in the blue or the purple line, those individuals had a higher mean arterial pressure maintained over the first 7 to 20 days after injury, suggesting perhaps that higher blood pressure in the acute setting is beneficial through driving increased perfusion to the spinal cord. And again, this is just really quantified in a slightly different way here. These columns on the right are for cervical SCI and for individuals with motor complete and incomplete injury. And again, you can see here quite nicely, those that had the greatest improvement on the right here had the highest average blood pressures during the acute hemodynamic management period. So this really raises the question, well, okay, maybe blood pressure augmentation is good in the acute setting, but if you increase blood pressure too much, there is definitely a chance that some of the damage caused by injury where blood vessels are disrupted may result in increased bleeding in the spinal cord. That's very hard to assess clinically. So there's been some studies that have looked at this in the large animal model. And I'm going to share some data here from my collaborator, Brian Kwan's lab. And what they essentially did is exposed pigs to a spinal cord injury, and then they looked at vasopressor administration. And they gave the spinal cord injury here. They provided compression of the spinal cord for the first 2 hours and then decompressed for the remaining 2 hours. And then pigs were exposed to one of 3 or one of 4 conditions, either no hemodynamic management. They used pressure augmentation with vasopressors during compression or during decompression or during both compression and decompression. And this probably represents, clinically what happens most often, because decompression is commonly very delayed in people with spinal cord injury. And then basically if they look histologically in the spinal cord, what you can see is indeed, if you do provide pressure augmentation in the very acute setting during compression and decompression, there is increased hemorrhaging within the spinal cord. And increased hemorrhaging is certainly not a desirable outcome. It creates a lot of cytotoxicity and also produces or starts off many cellular cascades in the injury, which we think may exacerbate the secondary injury. But this really got us thinking, blood pressure is, made up of a few different things. And if you go back to some of the classic kind of measures. We know mean arterial pressure is cardiac output times total peripheral resistance, and cardiac output is made up of stroke volume and heart rate. Since we now know from our rodent models that heart function is impaired almost immediately after injury, it got us raising the question, well, what if we target the heart as a way to improve hemodynamic management? And so this is now some of the work from our lab and postdoc Alex Williams. Essentially, the question that she posed is now to use our pig model of high thoracic spinal cord injury. We can give them the injury. We then start one of 4 different types of hemodynamic management. They either get no management, they provided norepinephrine infusion across this compression and decompression period. So again, this is kind of similar to what you saw in the previous slide and the clinical norm. Uniquely to this study, we also used dobutamine infusions. Now, dobutamine will target the reduction in cardiac contractility directly. Norepinephrine will, of course, act in the periphery. And so whilst both of these probably increase blood pressure, they do so by different mechanisms. And then in this model, we were able to fully instrument the heart. So again, we're using a left ventricular pressure volume catheter to generate LV PV loops. We also have a pulmonary artery thermodilution set up to enable us to measure direct cardiac output and make sure we can validate the volume measurements coming off the LV PV probe. And then we have an inferior vena cava occluder catheter put in as well to make our measures of end-systolic elastance. I'm first going to share just the data to demonstrate the efficacy of these hemodynamic management approaches. So orange is norepinephrine, green is the high-dose dobutamine. And what we actually found is both norepinephrine and dobutamine do a good job of increasing cardiac contractility and blood pressure. But what we're also able to do in the pig spinal cord is actually instrument and make direct measurements of intraparenchymal blood flow, oxygenation, and metabolism through microdialysis. We do this with probes inserted directly below the injury site at varying distances. And what I'm sharing here is just some of our spinal cord oxygenation data. What you can clearly see is despite similar responses on the hemodynamic management side, it's really only the dobutamine that is able to normalize spinal cord oxygenation. Now, of course, because blood pressure wasn't different between these two groups, it does raise the question, well, did dobutamine normalize the hemorrhage? And really, I've got a mixed bag of results to share with you here. If we look at the hemorrhage from our control, so this is a longitudinal version of the spinal cord. This is with a dobutamine-treated animal, and this is a norepinephrine-treated animal, and then this is a cross-section through the injury site. What you can clearly see is there's definitely still hemorrhage associated with the dobutamine because blood pressure is up. It's maybe not as severe as the norepinephrine, but it's certainly still more present than the controls. And so what we're now doing in the lab is trying to think of combinational approaches that can use, this perhaps better hemodynamic management strategy, but pair it with a therapy that targets directly in the spinal cord. And this will be the work of Ryan Hoyland, who's now a postdoc in the lab. And we're basically looking at ways we can try and promote blood flow to intact vessels within the spinal cord through manipulating spinal cord vascular resistance. So that's what I have to share with you on the acute side. I'm going to switch now really on the last part of this seminar almost to talk about some of the interventions for the chronic setting. As I mentioned in the introduction, there's about 2.5 million people living with spinal cord injury. And obviously, they've now had their injury for many years. We've got very good at managing these individuals. And so they're now living longer than ever before. And what we're actually finding in these individuals is that cardiovascular function is now emerging as the primary cause of mortality in these individuals. So there is now some research trying to optimize cardiovascular function in this chronic setting. And I'm really going to share with you some of the work we've been doing in the lab around 2 different therapies. The first of those is intermittent hypoxia. Now, again, this is the chronic setting, not acute setting. And intermittent hypoxia is this emerging therapy for the field. Basically, it describes the delivery of short periods of low inspired oxygen interspersed with normal oxygen levels. What has been shown in other systems is that this type of intervention, intermittent hypoxia, elicits plasticity through the carotid body that results in long-term facilitation within various motor pools. This has been studied across animals and humans, and that plasticity within those motor pools and long-term facilitation has been shown to result in everything from improved tongue and airway function. A lot of work has been done on breathing function, and more recently now on forelimb function in the rodents, and also lower limb locomotor function in humans. Of course, intermittent hypoxia is also a potent stimulus for the cardiovascular system. As the carotid body is exposed to low levels of PO2, this will stimulate the chemoreflex, which will activate sympathetic circuitry to elicit changes in cardiac contractility and blood pressure. So with this idea of long-term facilitation in neural pathways being a common finding from intermittent hypoxia, and us knowing that it's really a reduction in the sympathetic pathways that's driving the cardiovascular dysfunction after injury, it really led us to the question, well, can we use intermittent hypoxia to activate or increase activity and facilitate these sympathetic pathways that are spared around the injury site and normalize cardiac and vascular function? So this is some very recent research in the lab. I'll just share with you what happens to sympathetic nerve activity. So outside of the field of spinal cord injury, intermittent hypoxia is used as a way to mimic sleep apnea. And it's certainly been shown that if you make just sympathetic nerve activity recordings in response to multiple episodes of intermittent hypoxia, so here these peaks are each bout of the hypoxia, what you see is this gradual increase in sympathetic nerve activity that continues when the delivery of hypoxia is ceased. And we term this increase in activity long-term facilitation. And it's thought in the field of sleep apnea that this long-term facilitation sets the stage for some of the hypertension and cardiovascular remodeling. Of course, in spinal cord injury, we suffer the opposite. We suffer from low sympathetic nerve activity. So what happens to sympathetic nerve activity in a rat with spinal cord injury when exposed to hypoxia? Well, some work by Mehdi in the lab and Lisa, two really outstanding PhD students that have really been investigating this therapy. We've been able to essentially show the same thing happens in the sympathetic circuitry. So remember, again, this is a contusion injury we're inducing. There are some pathways spared below around the injury site. And what we've been able to show is that intermittent hypoxia can indeed facilitate activity in these pathways. If we take a look at what happens to the heart and blood pressure at the end, what we're showing here, and I appreciate this is a little bit of a busy figure, but the black bars overlaid with the individual rat responses represent our spinal cord injured rats. The first one will be baseline, and then we go to 30, 60, and 90 minutes post the intervention. The green is the uninjured rats, and what you can see across all of these indices is that there's really no effect of this intermittent hypoxia on any indices of cardiac or vascular function in the uninjured rats. But in the injured, the spinal cord injured rats, we see a really nice increase in practically every metric of left ventricular pressure function we take, or indeed just in blood pressure. It still doesn't quite get back to normalizing the function to an injured rat, but it certainly increases function. And this persists and peaks at around 90 minutes post-delivery. And again, the way we think this is working is by activating some of that spared circuitry. There's been another really prominent field of research in spinal cord injury around the use of epidural stimulation. So stimulation here describes now providing an array which is implanted surgically and epidurally. And basically what this would enable you to do through a series of anodes and cathodes is provide stimulation to the circuitry below the injury that is perhaps disconnected from the supraspinal structures, or at least has reduced activity. So it's a way to kind of boost activity. Some really great research now from Aaron Phillips' lab in Calgary has shown some of the mechanisms by which this works. And the way it works is if you put this stimulator on the dorsal surface of the spinal cord, then it stimulates the dorsal root afferents, which then activates sympathetic preganglionic neurons to increase blood pressure, and now we're showing cardiac function. This is highlighted here really in a case study that Dr. Aaron Phillips and I worked on a few years back. What we did here is to take an individual with spinal cord injury. We had them laying down. We then moved them to the sitting up position and measured their cardiac function. We see this nice reduction in end diastolic volume, stroke volume, and cardiac output. When we do this with a stimulator on, you can see we can abrogate or reduce the magnitude of drop in these indices. Some work now on the experimental side through a large collaborative project is now showing the same in animal models. So here we've got left ventricular pressure and volume, gradually increasing stimulation amplitude on the bottom here. And what you can see is if you take the pressure volume loop here or with stimulation, there's really quite a profound increase in cardiac pressures, almost normalizing cardiac pressure with epidural stimulation. So with that, I'll kind of just summarize where we're at and really share one slide with where I see the future going for cardiovascular research. So on the animal side, there's still plenty of work to do. One of the things we're working out in the lab is trying to link anatomical and functional plasticity in the sympathetic circuits. There's also a lot of research to be done on mechanisms of axonal degeneration and regeneration, particularly in the autonomic nervous system. The bolded ones, by the way, are things we have actively going in the lab. In this acute setting, again, we're really working towards trying to optimize cord oxygenation and reduce secondary injury. Many other groups now are working on cell therapies and scaffolds to transpose the injury site. And then I think in the chronic settings, an area where there's really a lot still to be learned. So there's many interventions now that are being used, from stimulation to hypoxia. I didn't talk today at all due to time on activity-based therapy, but there's a lot of work to be done there. And certainly some of the ways we and others are thinking is to start to combine some of these neuromodulation techniques with activity-based therapy and look at these combinational approaches. And of course, anything that involves exercise or changing physical activity levels, there's a fair amount to be done around changing those behaviors as well. One of the other potential limitations to some of the work I've shared today is that many of the animal experiments that we've been conducting have been done under anesthesia. And that's really because we've been limited in the field to be able to make these measures of pressure volume loops in awake animals. But certainly with some of the new technology that is now available, I'm really excited to work with Translational on some of these initiatives to utilize telemetric devices to be able to make some of these measures in awake animals and remove, I'll say, the potential confounding impact of anesthesia. I should have said all of our anesthesia that we used and I showed here was done with urethane in the rat models. So it does do a pretty good job of maintaining sympathetic vasomotor tone, but there is still anesthesia there. So we think this has some really good applications in the future. With that, I'll just bring together some conclusions. So hopefully I've been able to share some data today from my lab that has demonstrated that spinal cord injury really impairs this bulbar spinal control of sympathetic preganglionic neurons, that we're showing has some really profound downstream impacts on the heart and vasculature. In the acute setting, it seems that targeting the heart may offer a better alternative for hemodynamic management, and we're now working towards trying to manipulate the vasculature in the spinal cord further. In the chronic setting, certainly the most promising therapies seem to be ones that target this reduction in activity in the sympathetic circuitry. And two of the most promising, at least in my mind, seem to be intermittent hypoxia and or epidural or some form of stimulation to the spinal cord, both of which appear capable of increasing cardiac function and blood pressure. And then future studies, I think there's a big space here for these combinational strategies to try and improve cardiovascular outcomes for the field. With that, I would like to mention a special shout out. I've had, been very fortunate along the way to have wonderful mentors that have really helped me in setting up and supported this program of research, both logistically and content-wise as well. And really wonderful group of collaborators that have made expanding into some of these new areas available, and of course the funding as well. And with that, I would like to thank you for your attention and happy to also answer any questions as well. Thank you.
Thank you so much for that, Chris. Really fantastic and such a great way to kick off the series. Okay, so the first question here is, have you evaluated functional/physical changes in the intracardiac nervous system?
Yeah, that's a really fascinating question, and one that certainly there's some really interesting research coming actually out of Italy that has shown that the sympathetic nervous system plays a really important role in tuning cardiomyocytes, and that has been of interest to me. I have not done that as of yet, but it's a fascinating area. to my knowledge, no one has looked at that in the setting of spinal cord injury or indeed following any injury to the central nervous system. I think the only research on that has been where studies have pharmacologically blocked the sympathetic nervous system to look at that. But yeah, absolutely on the list of things to do. So really great question.
Awesome. Next question here is, with spinal cord injury and loss of central sympathetic drive, have you assessed a rhythmic potential in your models?
A rhythmic potential? I just want to make sure I understand that question correctly. So I'm not sure.
I think they may just be referring to if the potential for an arrhythmia to develop has been assessed.
Yeah, so we have looked at arrhythmias, and there's certainly as many other studies that have shown that spinal cord injury is associated with many different forms of arrhythmia. It's also been shown that if you give sympathoagonists, that there is an increased prevalence of arrhythmia. We've also used telemetry devices to look at circadian rhythms, which is potentially where this question is asking as well. And what we tend to find is that when injury is high, both in individuals and in fact in our rodent model, circadian rhythms, at least in things like heart rate, body temperature, and blood pressure, are lost. Interestingly, in the rat model, some of those appear to come back, but I'm not yet aware of any studies. Many of those are thought to be linked to changes in sympathetic nerve activity, and I'm not aware of any studies yet that have utilized long-term awake measures of telemetry to measure sympathetic nerve activity and look for circadian oscillations in that too. But certainly, yes, and in people from 24-hour blood pressure and heart rate recordings, we do know that when the injury is up in the cervical cord, there is a loss of circadian rhythms.
Okay, fantastic. The next question here, I think we'll kind of branch off of that a little bit. This person has asked, is there any consideration for using rodent models for spinal cord injury cardiovascular assessments, given that rodents are or tend to be more sympathetically driven than large animals and humans, which are more parasympathetically driven?
Yeah, and this has always been on the back of my mind, and partly some of the limitations, it's obviously not just in autonomic balance, it's also in size of the heart as well, and even just in heart rate versus stroke volume relationships. In the rat, the heart is beating in our studies around 450 times a minute. That's clearly not the case in humans. And so this was really what stimulated the collaboration with Dr. Brian Kwan. He had this pig model of low thoracic injury to look at motor and sensory outcomes. But of course, as many people on this line know, the pig is also a fantastic model for the cardiovascular system as well. And so that was part of what prompted the move to do some of the research in the pig model as well. I will say that at least from the data we've collected, the magnitude of reduction in things like cardiac volumes and pressure volume related indices actually appears pretty similar across both rats and pigs, which gives us some confidence in at least extrapolating some of those functional measures across. But the point absolutely holds, and we've certainly seen that when we've been doing some blockades on both just the parasympathetic and sympathetic side of things. So it's definitely a consideration. To put it into perspective, if we want to run a pig study, and the way we do that at UBC through our Center for Comparative Medicine, it costs us about $10,000 a day to do those studies. So utilizing the large animal model, and there's definitely some more ethical considerations with continuing to use large animal models as well. So now we've shown kind of this similar reduction across both. We tend to use the small animals for a lot of our long-term studies, just from a cost and ethics perspective, and really save the large animal work for the more clinically relevant questions, such as the hemodynamic management question, where we can also instrument the spinal cord as well as the cardiovascular system. So always a consideration, and yes, absolutely.
Awesome. Thanks for that answer. Next question here. What was the quote unquote dosage of hypoxia that was used in the study that you mentioned about intermittent hypoxia?
Yeah, so the one we've settled on, and we've tested a few different ones. So I don't know if you're familiar on the spinal cord injury side or the sleep apnea side of things, but we've settled on 10 episodes of 1 minute of hypoxia, which is delivered at 10% fraction of inspired oxygen. Interspersed, we actually interspersed with 100% oxygen in our anesthetized preparations. We are now doing some long-term work with intermittent hypoxia. That's some of the work from Lisa, a PhD student in the lab. And there we do a similar protocol, but rather than interspersing with 100% O2, we're interspersing with room air O2. We have tried some of the other protocols. In spinal cord injury, one of the most common protocols is a 3 by 5 minute. But in our level of injury model, that the animals don't do too well on that from a blood pressure perspective. It causes really profound reductions in blood pressure. And we certainly steer clear of the more severe hypoxia with many exposures like they use in the sleep apnea field. So kind of somewhere in between those two models, 10 by 1 minute of 10% fraction of inspired O2. I should say as well that we do collect arterial blood samples throughout and run arterial blood gas analysis. So we are careful to make sure our carbon dioxide is staying consistent throughout the exposure and into the post-hypoxia recovery period as well.
Fantastic. Thanks for clarifying that. Next question here. For your pressure volume data collection, have you done any work on the right side of the heart? And what effects have you seen there in terms of contractility?
That's a really great question. And, a shout out to anyone here that is developing technology on this side of things is, it can be really challenging to do right-sided pressure volume loops in a closed chest approach. What I mean by a closed chest approach is, keeping the thoracic cavity closed. So for all of our measures, we go in through the carotid artery and down into the LV. We have done some right-sided pressure volume loops. And in fact, one of my PhD students presented some work at experimental biology doing biventricular pressure volume loops, where we get PV loops from the right and left side of the heart synchronously. I will say getting the right-sided pressure volume loops is quite a challenge. It tends to work better in scenarios where the right ventricle becomes dilated. In ours, the right ventricle is becoming smaller. We've not yet got anywhere near a sufficient amount of animals with spinal cord injury on the right side of the heart to be able to answer that question. I will say we've looked histologically at the cardiomyocytes on the right side of the heart, and we see a similar degree of atrophy on the right versus the left side. So we suspect that what is happening on the left is happening on the right. But we're trying to get to that answer. We haven't yet done open chest pressure volume loops because there are also changes in lung function and pulmonary pressures after injury, and we try our best to maintain those around the heart to keep things as, physiologically as possible. But yeah, really fantastic question.
Awesome. So I have two questions to follow up. One is about cardiomyocytes and one is about lung function. So the first one is, do you measure lung function in this model?
We have not measured lung function in this model. I would have no reason to believe that you get much of a reduction in lung function with this model. The diaphragm certainly is very well preserved. I'm assuming they mean in the rat model. We've not done it in the rat model. The expiratory muscles, of course, will have lost supraspinal control. But again, from a lung function perspective, most rat studies use a cervical hemisection model to look at lung function responses. I do believe some people have tried this with a similar high thoracic model, but there wasn't really any marked reductions in lung function. Interestingly, on the human side of things, where I've measured lung function a lot in individuals with cervical spinal cord injury, we do see quite a profound reduction in vital capacity. Total lung capacity tends to be actually relatively well preserved. So it's really anywhere where there's an expiratory, where expiration contributes to the measurement of lung function. So things like vital capacity, expiratory mouth pressures, or expiratory reserve volume are already compromised in individuals with cervical spinal cord injury. And that reduction in lung function actually has some really interesting effects from a heart-lung perspective. And certainly we've been looking at ways on the human side of things to be able to manipulate the respiratory system through changing pressures to be able to promote left ventricular function as well. So yeah, really great set of questions. It's not something we routinely look at in the rat, but definitely in the humans we do.
Awesome. Okay, the next question here I mentioned was about cardiomyocytes. So this person has asked, have you measured cardiomyocytes following intermittent hypoxia? Since all tissues are exposed to fluctuating patterns of oxygen, could intermittent hypoxia elicit benefits in the periphery in addition to activating neural circuitry?
They absolutely could, and that is exactly, we have the tissue now to be able to answer that question, but we have only just finished running those long-term intervention studies with the intermittent hypoxia. So, and we have more that are ongoing in the next couple of months. So we're not yet in a position to answer that, but we've absolutely got some tissue ready to answer that question, and we'll be answering that in the future. So yeah, great question.
Fantastic. Awesome. Okay. Next question here. Have you tested dobutamine effects in your chronic rat model of spinal cord injury, and do the hearts still have the capacity to increase contractility?
Yes, that is a great question as well. We have tested it. We've done it in a few different ways. We probably some of the easiest and most straightforward is where we've just done dose response. Dobutamine infusions in chronic rats. We did this almost to try and mimic an exercise response. A lot of people use dobutamine for that reason. And what we actually find is, yes, indeed, the heart can still mount a contractile response. Actually, for a given dose of dobutamine, at least at the low dose end, the cardiac beta receptors seem to almost hyper respond or over respond to that stimulus. Whether the magnitude is normal, is the increase with dobutamine the same as in an uninjured rat? We've not yet answered that in the rats, but we have answered that in the pig model. And in the pig model, at least, if you go 12 weeks out after injury and you measure end-systolic elastance pre and post dobutamine infusion, then the heart reduces, there is a reduced maximal pressure generating capacity in the chronic pig setting. We've been working on some ways to try and normalize that with different hemodynamic management strategies, but yes, in the pigs that happens. In the rat, at least what we've seen so far, that doesn't seem to happen to the same degree, albeit that we've not been able to measure end-systolic elastance pre and post dobutamine infusions in the rat. That is extremely technically challenging to do, because as you deliver the dobutamine, you tend to get a bit of a peaking on the upper left corner of your pressure volume loop due to the increase in contractility. And that can confound some of your measurements of end-systolic elastance. So it makes it technically challenging to do. The pig doesn't have that same limitation. So it's actually an easier question to answer there.
Fantastic. I did not realize the time, so that's going to be our last question. But I want to thank you so much for your fantastic insights today. It's really been a pleasure to have you with us, and I hope that you had fun.
Yes, thanks so much for having me, and thanks to the audience for the excellent questions as well. Thank you.
Great. And I also want to send a very big thank you to our audience for joining us live. In closing, we hope that you enjoyed this Inside Scientific webinar sponsored by Translational and produced in partnership with the American Physiological Society and the European Council for Cardiovascular Research. And we look forward to having you with us for the rest of the series. Thanks again.